My name is Tim Child. I'm Executive Director of the University of Chicago Alumni Association, and it's a real pleasure to be here with you today in Atlanta and to welcome you to this Chicago Society Insights event. And I'm here really for two reasons. One is to thank you for your support of the institution and talk a little bit about what the Chicago Society is. And the second is to provide you an opportunity in the person of Milan Mrksich to get a vivid demonstration of why that support is so important to the university. I don't know if any of you read in the New York Times over on Sunday, there was a review of a new book out by Jonathan Cole, who is the provost at Columbia University and not otherwise associated with the University of Chicago. The name of the book is The Great American University, Its Rise to Preeminence, Its Indispensable National Role, and Why It Must Be Protected. So it's a lot to the title. In it, he says, I'll just give you a little quote at the end here and read two things. The University of Chicago is, in his contention, quote, our closest approximation to the idea of a great university. It is a meritocracy of ideas, a place where ideas flourish in an open way. Now, we say that about ourselves all the time, but it's always nice to hear it from an outside source. It's corroboration, I think, of what we hope and believe about the University of Chicago. I think the Chicago Society, which is a group of supporters of the university who give $2,500 or more a year to the university, to any part of the university. I myself give to the business school where I have a degree, give to the divinity school, and I give to the medical center. I think I'm just trying to cover all my bases. But it's really any aggregation of gifts or single gifts that people make of $2,500 or more to the university, and about half the cash the university receives each year comes from Chicago Society members. So it's a very important group of people, some of whom are in the audience, and we want to thank you for doing that, for making those gifts. But we also want to encourage everyone who's here to give and make the university a part of their philanthropy. The why part, I think, is also easy, and Jeff's going to talk a little bit about that in a moment. But I wanted to also talk about one last bit from this article in the Times about the why. And the author says here that when we think of these universities, we don't think that lasers, FM radio, magnetic resonance imaging, global positioning systems, barcodes, transistors, improved weather forecasting, methods of surveying public opinion all had their origins at our research universities. Read the book and you will learn that the algorithm for Google searches, DNA fingerprinting, fetal monitoring, scientific cattle breeding, and even Viagra are the results of university research. So I think if you agree with all of us here that are from the university and those who support the university, these research institutions are absolutely vital, not just because of the knowledge and the boundaries of knowledge that they pursue, but how that affects our business, our economy, and the quality of life. So I want to thank you very much for being here, and I want to introduce Jeff Kobler, who's the chair of the Atlanta Chicago Society Committee, who will introduce our speaker.
I'm Jeff Kobler, Booth School of Business in 03. On behalf of the University of Chicago, I say thank you to all of the Chicago Society members and guests for your continued support. And like all of you here tonight, I wanted to share why I chose to attend the University of Chicago. I think we all chose to attend for the same reason, although I didn't realize it at the time. And that is the repercussions of research in any one college prepare us to lead in our own professions better than any other institution. When Chicago research advances in, say, chemistry, its implications are not constrained just to chemistry, but also influence business, law, healthcare, government, and other fields. I chose to serve as a volunteer for the Chicago Society because I believe in the power of that interconnectedness. because of the research, teaching, and scholarship that it supports. So if you're interested, please join me and ask either myself, Tim, or Emily Bauman after the presentation tonight. There are many ways to give and volunteer. I would also like to recognize committee members who are in attendance and ask them to briefly stand as I call their name. David Christopher, MBA 98. Elizabeth Rogan, AB 81. And Mason Stevenson, JD 71. Thank you each for your commitment and participation on the committee. Now it's my honor to introduce Professor Milan Mrksich. I am pleased to introduce him. He's a professor of chemistry at the University of Chicago, an investigator with the Howard Hughes Medical Institute. Professor Mrksich is an active advisor in government and industry, currently serving as chair of the Defense Sciences Research Council, an advisory group for the Defense Department, and the Board of Governors for Argonne National Laboratory. He's also founded and serves as an advisor to several biotechnology startup companies. Professor Mrksich is a leader in the science and application of materials that contact biological environments, and his laboratory has pioneered several technologies, including strategies to integrate living cells with microelectronic devices, methods to enable high-throughput assays for drug discovery, and model systems for understanding the interactions of cells with their tissue environments. But I see you all sitting there, and I know you're all asking yourselves the same question that I did after reading that. How do cells control the assembly of focal adhesions that mediate cell adhesion to extracellular matrix? And clearly, that's the overarching question. Fortunately, our speaker can answer that question right after he discusses his recent work that is developing the molecular engineering tools to identify and model the molecular mysteries of the cell and to engineer cells with novel functions. Please join me in welcoming Professor Milan Mrksich.
One, my wife is absolutely unimpressed with that list of scientific accomplishments. And two, my mother-in-law is absolutely unimpressed with my last name, which she still can't pronounce right. So you did a great job with that, Jeff. So I'd like to also thank Jeff and Tim and Emily for organizing the event, and it's great to see everybody come out. I've done a couple of these over the past few years and always enjoy it a lot. It's you can't find a more interesting group of people to spend an evening with, and for us scientists, any chance you give us to talk about our work, we rapidly accept. So on both counts, I'm glad to be here. The broad theme that we've been thinking about in the university is one in molecular engineering. And as you know, President Zimmer has been advancing us towards a start of a molecular engineering initiative in the form of an institute. And that's made a lot of people stop and think, what is molecular engineering? We know what electrical engineering is and mechanical engineering and other forms, but there isn't around the country yet a department of molecular engineering. And as he's developed his vision, and I think many of us have subscribed to it and in fact taken a lot of motivation from it, we find that it's quite natural to think about molecular engineering. The engineering disciplines themselves over the years are always striving to do things at a smaller and smaller length scale. Can you make a transistor in a computer chip smaller so that you can get more transistors on board and do more computation? Instead of moving chemicals through large pipes in a chemical plant, can you actually put a chemical plant on a chip and come up with tiny little pipes or microfluidics? Can we create airplanes that are the size of a mosquito so that they can hover around and collect basically intelligence? So a theme in engineering has been smaller and smaller, and as you go smaller, eventually you start to bump into molecules. The matter doesn't look like bulk matter that you might look at, but in fact something very different. And it's chemists and physicists and biologists who in fact worry about molecules in that context. And so the engineers are in fact losing their ability to manipulate matter, control matter at that length scale, and it's quite natural in fact to think about an enterprise of molecular engineering that goes beyond the traditional engineering and brings these scientists where Chicago has such strengths to bear. And so that broad theme, we can talk about this a bit more at the end. What I wanted to do tonight is give a 30-minute or so discussion about one example of a molecular engineering problem. It's one that we started thinking about several years ago, and it's forced us to think differently about how cells work, and it's forced us to think differently about creating the tools that can unlock the mysteries of the cell. So let me jump into the story. I'm not going to make this a long or too deep lecture. I have to give an organic chemistry lecture tomorrow morning back on campus, so I'll save that pain for that group of students. You don't deserve that. And let me start here. Let me talk about design and systems. So engineers have come up with just amazing examples of devices that are complex, and those include, I don't know if we can get the lights down just a little bit on the stage so that the slide is easier to see. But those include the new Dreamliner from Boeing, computer chips, analog devices, etc. And the thing that we know intuitively is when we design something in the material world, we actually have a blueprint that describes how it's put together and how it works. I mean, we have to have a blueprint because these are man-made devices. And blueprints are important because they tell us how to put the device together. When it breaks, you open up the blueprint and you figure out how to fix the device, whether it's a car or a computer chip in some cases. They tell us how to predict failures because we know when different components are likely to stop working. And they actually help us change the function. If you want to swap out one part of your engine to put in something that will make your car go faster, you know how to do that according to the plans. The cell in the upper right is also an amazing machine, as I'll show you. And it's one that we, in biology, have been trying to sort out the blueprints for. We want to understand how the cell works. But it's one where we still can't claim success. We still fall short of having what you might call a blueprint for the cell. And let me give you a few examples why I say that. About a decade ago, there was a lot of fanfare and excitement over the sequencing of the human genome. We finally had the complete DNA sequence for us. And that was meant to give us the instruction set, the blueprint. It was meant to tell us what we're made out of and how we work. But in fact, while it's given us a lot of insights into medicine and biology, if anything, it's given us a good recognition that many of our assumptions of how the cell works have been wrong. It's taught us more about what we don't know than what we do know, and that's frequently what happens in science. But a few examples. Drug development. We still do it by trial and error. When Merck wants to come up with a new cancer treatment for a specific type of cancer, what they'll do is set up a little experimental test that kind of captures what's going wrong in the cancer, and then they'll run that test 1,000,000 times, each time in the presence of a different small molecule. And they're just looking by chance to see which of those small molecules happen to stop the bad activity. And that becomes a hit, a drug lead. So we're not designing drugs. We're just doing experiments really, really fast so that we can find by accident things that might work. Diagnosing disease. I just had to go for a physical with a full blood test. I don't want to get into that story because it's a messy one. But the point is, it's pretty amazing how many things are measured in blood. But it's also pretty amazing how many diseases can't be diagnosed efficiently. And you have to go back and you try something, and you go back in a month and you try something else. And it's a pretty messy process. It turns out that biomarkers, the things we measure in blood, like PSA, the test for prostate cancer, the FDA approves only about one biomarker a year. That's hideous. The NIH spends $30 billion a year on basic research. Some fraction of that money is directed towards developing ideas and then validating biomarkers. Yet, we're only finding one a year. Something's wrong. We don't understand disease at that most basic level. And then the last one, predicting failure. And what I mean to say here is, we're made up, as you'll see in a moment, of about 100,000 proteins. They're all essential, otherwise we wouldn't need them. Evolution would have gotten rid of them earlier. But we don't know what most of those do, what 90% do. and we can't predict what they do. The way we find out is we take a mouse and we genetically engineer it to either put in a human protein or take out one of the proteins, and we see what happens to the mouse. In this case, the little mouse is the normal guy. The big mouse was genetically engineered so that it makes human growth hormone, and as a result, it grows bigger. But that's how we go about figuring out what the part does. Imagine a Boeing 787 where we don't know what the parts do. The only way you have to figure it out is pull the part out and then send the thing in the air. That's how we do biology, okay? I've got to be careful. There's a hole cut out here, and I don't like to stay still, so should have covered that with the podium. So there is a blueprint of biology. There are rules out there. It's a rational thing. How do we figure it out? We go down to the basic living unit, which is the cell. All of our tissues are really just collections of cells, and the cell is the minimal living unit. It can grow and culture, it can reproduce and divide, and it has many of the aspects of something living. Smaller than the cell, you're just talking about molecules in a tube. And we know a lot about the cell. It's about 50 microns in size, so that's a width of half of your hair, for example. We know that the parts list is about 3 billion bases of DNA. 3 billion isn't a big number anymore. We all have 3 gigabytes of memory on our computer, so all of you can walk around with your DNA sequence in your pocket. That's not bad. That DNA produces about 100,000 proteins, plus or minus a factor of 2. 100,000 isn't a big number. There are machines that have more parts than that. And about 100,000 metabolites, things like glucose and other things running around the cell. And then a little bit of water and salt and lipid. So the parts list isn't daunting. It's something we can get our hands on. But what makes the cell complicated is that on the right, all these molecules have very unusual distributions in the cell spatially. So that unlike having a can of juice where everything is present in equal concentration throughout the sample, a cell organizes where the components are. Now that's not unusual on its own. What is that within the cell, the locations of these molecules are constantly changing to bring them to the site where they're needed to do something. It's kind of a on-demand activity. And to give you a sense for that, there's a beautiful animation that was prepared a couple of years ago that gives you a movie of looking inside the cell and seeing these protein molecules interact with one another and move around and form structures and break those structures down. So I'm going to be quiet for a couple minutes here and let you watch this. If you were to look at a computer chip, nothing would be moving. But this is how the cell carries out functions. And for those of you who, some of you might have recognized some pieces, what's amazing to me is that this animation is very faithful to the actual science and what we know about cell function. So you saw some images of these tubes being formed and then other tubes being broken down. A cell has a skeleton, a mechanical structure. And when a cell crawls, it has to keep remodeling that structure. It's building the structure at the front end and degrading it at the back end. And that way it can move along a surface. Now that activity is critical to cancer because metastasis, the spread of cancer, requires that a tumor cell crawl out of the primary tumor, get into the blood vessel, and then circulate where it might find another home. So understanding how those structures form and degrade give us insight in a different strategy to treat cancer, to prevent that kind of remodeling. But the beauty there is that within the cell, these molecules are almost magically coming together to build machines where they're needed, when they're needed, and when they're not needed any longer, they just fall apart and the pieces go back in the pool until they're needed somewhere else. So it's really a remarkable design. Now, let me give you a couple of examples of what kind of properties stem from this kind of architecture, if you like. And I think of these as living devices, and I would compare them to basically computer chips, semiconductor devices. So I can barely read this myself. The top says that living devices are robust. They fail gracefully and they operate with imperfect components. Let me put it this way. A computer chip might have 500 million transistors. Every transistor is identical. It has an IV curve, which is the same as any other one. So if you swapped two, you wouldn't change the device. If some of those transistors had different properties, the chip wouldn't work. And so it's important we control it. Biology, if you look at the brain, every neuron is a little bit different than the next neuron. Its size, its response time, the magnitude of its signaling. Yet, the brain can work with these imperfect or crappy components and put them together to give a very robust design. If we knew how to do that in the engineering world, it would be amazing. We'd be able to start to push the limits because we could use components that we couldn't yet control well and make in big numbers so that they're all identical. Biology is adaptable. Our brains can very effectively learn to solve new problems. And you might have seen some of these examples of prosthetic devices. Soldiers who've come back without limbs get fitted with a prosthetic limb that literally plugs into nervous tissue so that the brain can learn how to control that limb again. Because the brain is plastic, it can take on new functions. Computer chips can't do that. We build them to do a job, and that's what they do until they stop working. And then the other one, which we always think of computers as being amazingly powerful because they're fast and they can do a lot of operations. But we don't realize that biological computers, the brain, are perhaps even more powerful, just at different kinds of problems. A computer can add a lot of numbers together and get the right answer. We can't do that. But we can look at a picture and in an instant find Waldo. And that's a problem that a machine can't solve. When we do automatic target recognition, for example, we have a heck of a time solving that problem with the computer. We're also good at working with systems where we have incomplete information and we can fill in the blanks. So if you see that movie on the right, just watch it.
Now we just thumb it. But of all the rides I've taken...
Okay, what's going to happen next? I mean, you know what's going to happen next. Well, did I get it?
In former years, we traveled incognito, then by freight. Now we just thumb it. But of all the rides I've taken, I like best...
Okay. So you saw that coming. And that's because we're able to take systems where we have incomplete data or project what's going to happen in ways that machines still have a very hard time doing. So my argument is that because these biological systems are wired differently, built differently, architected differently, they can do tasks that we still don't know how to do in engineered systems. So there's a real reason to go and try to understand what is this magic in a living system. How do the molecules come together? How are they regulated? How is the system designed? And if we could come up with a good understanding, we'd be able to apply that understanding to non-biological systems, engineered systems. So it would be a bio-inspired approach. So one more chart here to think about this. What is it that we think about when we try to key in on what's special about the biological system? And I'm not going to go through all of these. The first one I've already talked about, this idea of dynamic wiring. In a normal chip, once you make the connections between the different transistors, they never change. But in biology, we're changing connections all the time. And that's because this dynamic nature, and you saw it in that video. And that's true at all scales. Molecules within the cell, within the brain, synapses between different neurons are developing and fading. And that's a property we don't know how to work with yet in the material world. I think this third one is interesting, which is good enough performance. If you tell your computer to do the same task 100 times, some computation, it will give you the same answer 100 times. If you tell a biological computer to do a task 100 times, it will give you, in a technical sense, a different answer every time. But every answer will have the characteristic that it's good enough for the application. So that if I ask you to, in your mind, build a pattern, build an image, in order to recognize somebody. If you really go study that, you find that every time we'll build a slightly different image, but in every case it's good enough for us to answer the question. And that's a different way of thinking, and I'll come back to that. And then the last one I'll point to is this highly nonlinear behavior. I don't want to get too technical, but everything we engineer is based on linearity. And what I mean by that is when you turn your radio up, it's linear. If you turn the knob twice as far, the volume is twice as much. When you step on your accelerator, it's linear. If you press down more, you go faster. Imagine a system where you turned your knob up and the radio went louder by 100-fold. And then you turned it up a little bit more and the volume came crashing down. That's nonlinearity. That's how our cells work. That's how biology works. It's based on nonlinear components. The rule in engineering, every freshman learns this in electrical engineering, is engineer out the nonlinearities because they're too complicated. We don't know how to think about them. We don't know how to model them. So that's a key concept that, again, biology teaches us to look into and learn more about. Okay, I'm going to, in 3 slides, summarize 30 years of research in biology that has a very unhappy ending for a lot of biologists. And the idea is the following. If you study the cell as the basic unit, I almost stepped in there, then the basic problem is that the way you talk to the cell is you let a protein interact with something on the cell surface. And that sets off a series of events, protein activities, that ultimately change gene expression in the nucleus. So a cell is just sitting there, and it's waiting to be tickled in one way or another, and in response it changes its gene expression. And by changing its gene expression, it creates different proteins that can then be used to make these machines that do things. So we used to think that this was a pretty simple relay, that you'd have something bind outside at top, and then that would make the protein in the membrane do something that would recruit some proteins to the membrane that would become modified in some way, move down into the nucleus, bind to the DNA, and allow some gene to be expressed and eventually turned into protein. We thought it was a nice linear range. And we thought, you know, there are probably a few hundred of these linear channels that let a cell respond to a few hundred different signals. But then we looked a little bit more, and we started to see that if we were looking at one of these pathways, as we call them, there were other stimuli that could trigger the same response. So we started to learn that there was so-called crosstalk. This pathway and this pathway, which let the cell do 2 different things, weren't isolated, but there were some cases where one pathway could influence another one. Crosstalk, simple idea. And so we thought, okay, we're pretty smart because it's not as simple. It takes, you know, a little bit more thinking to kind of worry about these systems. These systems still have the characteristic that you can kind of keep track of every protein. That's what biologists are really good at. There might be 30 proteins involved, and they've got a good understanding of what each one's doing. Okay, then the problem was that we started to get better tools for characterizing protein activities, and we started to find that things are more interconnected than that, okay? It turns out everything is connected to everything else. So within the proteome, within these 100,000 proteins, on average, each protein will talk to or somehow influence several others. So this is a so-called protein interaction map. It's based on some good data, some flawed data. We haven't worked through all that yet. But it's basically trying to represent, if you put 30,000 proteins and then you draw a line between every 2 that can interact with one another, this is the map. So, and I note that these are 4 maps. One is for a frog egg, one is for a worm, one is for a fly, and one is for human, or we like to think of it as Clint Eastwood. And this is where you see that we're not all that different. And you've heard that in the sense that DNA sequence between different species is much more alike than it is different. Okay, this is the unhappy ending. What's starting to happen is we're recognizing that biology is becoming an information science. This data is really lots of information that tell us which proteins are present in a cell at a given state, what are the interactions, and once you get to this density of data, it's not possible to do an experiment, sit down at your desk as a graduate student, and analyze the result. You know, we're used to doing an experiment. You design the experiment, you go in the lab, you do it, you get a gel or some other kind of data as the output, you sit at your desk, you look at it for 10 minutes, maybe an hour, and you know what happened to your experiment. When you start getting data of this scale and you work with systems of this complexity, that's not possible anymore. Now you need to use computers and models and other things that biologists in general don't like. It's not an unfair generalization to say that one way undergraduates choose their majors is if they like math, they go into physics, and if they really don't like math, they might go into biology. Now that's, of course, not a fair generalization, but it has some basis in it. So what this means is we've got to think differently about how biology is done. And it's not just in recent times that biologists have had an aversion to math. This goes back to a famous naturalist 200 years ago. So you can read the quote there. Every attempt to employ mathematical methods in the study of biological questions must be considered profoundly irrational and contrary to the spirit of biology. And that was a widely embraced sentiment. And just to make sure that he wasn't misunderstood, you can read what he had to say to follow up. So my point is that the science is changing. It's becoming more of an engineering science, much more of an information science. And that has real implications for both education, how we train our students, how we retrain ourselves, but also for the kinds of opportunities that we'll be able to address with this science building. So I'm going to give you a little bit of a view now on how the tools have been changing to address these problems. Now, I've told you there are roughly 30,000 or so genes that can be expressed in our DNA. That means to say that this 3 billion base pairs of DNA has instruction sets for roughly 30,000, has roughly 30,000 instruction sets. And we'd like to know when we look at a cell, which of those 30,000 instruction sets is being used at any given time. Which of them is actually on so that they're making their protein. Because that tells you what a cell is doing. A kidney cell has a different pattern of gene expression than a liver cell does, and a cancerous cell has a different gene expression pattern than a normal healthy cell. So if we could see that, we could learn about the differences. And one way we do that is using so-called DNA chips, or gene chips. How many of you have heard about gene chips? Okay, so what happens is we make a surface and we tie down to that surface a piece of single-stranded DNA. If we take a cellular sample, if that gene is on, it's producing a single strand of RNA. And RNA and DNA will form a ladder. They'll pair with one another if their sequences are complementary. And so if a gene is being expressed, that RNA strand will bind to a surface that has the partner DNA strand. So if you make a surface that has a pattern of 30,000 different DNAs, you can literally look for the expression of every possible gene in one experiment. So this is an image on the upper right. It's about a square inch in size, and in that square inch are little circular islands, actually about 50,000 of them. Each of those has tied to it a different DNA strand. So now I can take my sample, apply it to the chip, and every one of those different circles is reporting on the state of a different gene. And so I can fingerprint the genetic expression profile of a cell using this tool. Now this is remarkable. This was created about a dozen years ago, commercialized, actually created about 20 years ago, commercialized about 10 years ago, and is used in every biology lab across the country, in academia, in companies. So this has been a revolution in experimental biology. You can now do thousands of experiments in the time it used to take us to do one experiment. That's unbelievable. And it's given us insight into cell function that we've not had before. So for example, we've got a systems biology department on campus that makes a lot of use of this tool. And you can do things like take cells, now from human, from different tissue, liver, muscle, brain, and you can collect those cells and analyze them to see what's the pattern of gene expression. And without going into lots of details, you instantly see that each of those different cell types has a different recognizable pattern. So that if I took a cell from one of your tissues, I can profile it and know what tissue it came out of. And you see where that goes. The same is true for diseased tissue, whether that's a bad kidney, cancer, perhaps Alzheimer-related impact on cell function. If we can use this kind of tool for diagnostics, that's going to be the highest resolution we would have in being able to understand the basis of that cell function. Now, let me go on. It's also the case, though, that the DNA doesn't tell us everything. So that what we kind of have in the central dogma is DNA, when it's being expressed, makes RNA, and the RNA is used to make protein. And the proteins are those molecules you saw in the movie that do the work. It turns out that protein function can be regulated in the absence of changes in DNA activity. And that's because proteins can be modified and can be turned on and off even while they don't change in the amount that's present. And so what's needed are tools that will let us do the same kind of experiment to analyze protein function. And that's where my lab has been a leader. We've been creating those tools to let us do high throughput profiling of protein activities in the cell. And again, without going in a lot of detail here, what we basically need to learn how to do is tie proteins or other biomolecules to a surface so that we can make an array of different molecules and then treat that surface with a biological sample, a cell lysate, as we say, and then be able to go in and look at those proteins to see which one of them have a binding partner that was present, which ones of those have been modified by an enzyme in the sample. And let's do this. One common example that I'll focus on here is kinases. So kinases are the enzymes that are involved in just about every cellular activity, and most cancers are caused by kinases being overexpressed. So have you heard of Gleevec, this kinase drug? Gleevec is a drug that derives from work at the University of Chicago, in fact, that binds to Abl kinase, and it turns out that a number of cancers, those cells are cancerous because they have high levels of Abl. Everything else in the cell looks like it's fine, but this one enzyme is present at elevated levels, and it drives cell proliferation. When that enzyme is at high level, cells divide. And think about it. If cells aren't aren't stopped from dividing, they keep dividing until the mass gets bigger and bigger and bigger. And that's what a tumor is. If those cells can spread into other sites, one cell can replant in another tissue. And if it doesn't have the ability to control its division, it starts dividing and dividing. And that's why cancer can be so difficult to diagnose, because you may have to find that one or two cells. That's a real needle in a haystack problem. And then it can be difficult to treat because you have to get every last cancerous cell to make sure that just one can't start the process over again. And that's why recurrence is such a problem. So how do we measure kinase activity? There are 500 different kinases in the body. That's why there are so many different kinds of cancer. Many of those, one of those goes off, and it can lead to a different form of cancer. Kinases have the job, basically, of adding a phosphate group to a protein. And so in the lab, we can create an assay where we create a surface, we learn how to tie down a protein fragment, and then we treat that sample with a kinase. I don't have a pointer here, that's why I can't show you exactly what I'm pointing at. But in blue on the right, that enzyme, that kinase, has attached a phosphate group to the peptide. So if you apply a sample to the chip, And after you rinse the sample off, you see a phosphate group present, then you know the kinase was active and present in that sample. That's the experiment. How do you see the phosphate group? It's not very easy to see. Normally what we do is we use radioactivity. There's an isotopic form of phosphorus which decays, it's radioactive, and so we do the experiment and then we measure how much radioactivity is left on the surface. That's why we faculty don't do research. We sit in offices and travel to events like this. But it's okay, we did it when we were in grad school. This is all safe. In this case, the assay, in fact, works pretty well. But I'll share a secret. There are lots of examples of enzyme activities, they're not all kinases, where we don't know how to perform the assay in the lab. And this relates to that problem that there are a lot of diseases that we don't know how to diagnose. And what can make it difficult is that you don't always have this option of labeling the product with a radio label, so that if the enzyme does a modification to the protein of a sort that doesn't let you incorporate the mass, the molecular weight of those molecules on the surface. So the idea is that if that peptide has been modified so that it now includes a phosphate group, the mass will go up. You see a spectrum on the right that's increased by 80 units. That reflects this change in mass because of the modification. And this was really important because all enzymes will act on their protein partners and effectively change their mass. With this technique, we could now look at every enzyme activity and have a very straightforward way of doing that and not be limited by the need to impart this label. And so this is a so-called label-free method. Once we did this, we could quickly do it on many assays. We don't have to worry about the details of those. And we could start to move towards systems where we might be able to create arrays where on one chip, on one fingernail-sized chip, we might have 100 different protein fragments. And if you take a cell lysate, apply it to that chip, each different cell will lead to a different pattern of peptide modification. We can read that with mass spec and have a way of, again, resolving what's happening in this complex mixture. Now, this is where it got fun for us because we had to do some engineering. We had to build some instruments, learn how to use robots, because we got to a point where we really wanted to do 10,000, then 50,000, then 100,000 experiments a day. Now, when I was in graduate school, if you did 5 experiments in a day, you felt pretty good. You had a beer that night, and you said, you know, don't push it. Don't go for the next one. It's been a perfect day. But today, students in my lab are literally doing 20, 30,000 experiments in a day, and it's mind-boggling. And we've had to learn how to do that ourselves because we've invented this new science that makes that possible. So this is an instrument. It's a commercial instrument that we've had to modify in a lot of ways, but that, for example, is a several $100,000 instrument. So my house is sitting there in one room in the lab. And this is an instrument that can record those mass spectra, can measure the molecular weights of everything on my surface. This is, on the lower right, an example of one of our plates. That's a glass plate about this size. And in the case that we're working now, it has 1,500 different spots on it. Now, it might be fun to kind of show you that at this scale, we can't do things with our hands. We can't do experiments that fast. So we have robots working for us all night and all day. And what you see here is a robot which is transferring solutions onto our plate. That plate has 384 different addresses on it, so we've done 384 different reactions. And this is literally the case, that we've got a room full of a few of these robots, and they can do the work of what would have been 100 students 2 decades ago. And again, we're still learning what that means, how to collect that data, how to analyze it. But let me let this one, make it look a little bit. It's kind of neat. These robots have 96 pipette tips, and they're able to pick up tips pick up little pieces of solution, a microliter apiece, and deposit those in little reaction wells, mix all of our experiments together, and then apply them to our chip like you saw in the previous clip. And they're really remarkable. They don't get mad. They don't yell at you. Once in a while they need to be reset. But the throughput that's possible is just something we couldn't dream of a decade ago. Okay, let me go on and now show you how the mass spectrometer works. So we take this plate and we stick it in the shelf of the instrument. The instrument sucks it in. It's all under high vacuum, and there are lasers inside that are basically blasting these molecules off of the surface. The molecules are charged, so they get accelerated down an electric field, and based on how long it takes for them to get to the detector, we know the mass. And so that's how we basically measure mass. And here what's happening is, this is in real time. Every 2.5 seconds, the instrument is measuring the result on a different experiment. So we can do about 3,000 experiments in an hour using this kind of approach. And what you see is this peak, this tall peak is present here. One experiment, it's not going to be present. Right there, okay? So what we've done in this was an experiment to do a screen to identify a drug lead for one of these targets. And what happens is we've got an enzyme which is modifying a protein by removing a piece of it. And so that peak on the right is being converted to the peak on the left. That's what the enzyme does. This is a so-called histone deacetylase. It's elevated in metabolic disorders, diabetes. And so we're interested in seeing if we can find a compound that binds that enzyme and stops its activity. And the hypothesis is that if we could stop that overactive enzyme, we might lead to a partial treatment for diabetes. And so in this experiment, in one day, we did 50,000 experiments where we repeated the same reaction 50,000 times, but each time we had a different small molecule, a different drug candidate present in the well. Now this is remarkable. Out of 50,000 compounds, 10 showed this kind of activity. They prevented the enzyme from processing that protein efficiently. So 10, that means 1 out of every 5,000. Now if you were doing experiments 5 or 10 in a day, you wouldn't find it. We could in one day do 50,000 experiments and quickly find the winners and then move on and validate those and test them out. So that really brings me now to the end of the story. I'm going to give a little wrap up. But the point here is that, well, let me just go to the wrap up and we'll do it that way. The point is, in the first case, that tools, technologies, are so critical to driving biological research. And if you, in this first point, that's the point. If you look back historically, one of the first tools in biology was the microscope. That's now 400 years old. But the microscope was one of the first tools because it let us see cells and understand that living inside of tissue are these cells that look like they're carrying out function. And then other microscopies came along. And then we developed a series of molecular tools, these abilities to analyze DNA function, analyze protein, create synthetic proteins so that we could test independently what a protein might do. You can take a mouse and you can put one of these human proteins into it and see if it drives cancer. And that then leads us to an understanding that that protein might be responsible in cancer, and then you go and follow that up. So the mouse experiment was a tool that was developed that had big impact. And there's a whole series of these. The tools over the past decade that many people have been excited about are the ones that let us look at cell function globally. And that's a real paradigm shift. We used to start asking a question by saying, this protein is what we're interested in. And then from there you build out. What's that protein interact with? What's that interact with? And so it's kind of like building a jigsaw puzzle. You start with one piece and you build off of it. The new paradigm is starting globally, not worrying about the details, not making an assumption about which protein is causing that disease, but just saying globally, I'm going to compare the proteins that are present in a non-diseased and a diseased sample. And then I'm just going to look for the differences. And then I'm going to follow up on those differences. And that's a different perspective because in the first case, you're going to be lucky that you picked the right one and then built out, or you're not. And statistically, you may be right 1 out of every 30 times. And so when the NIH funds research groups to do these kinds of things, they need to build in a lot of redundancy. So we all get money to work on the same problem, but I work on it from one perspective, you work on it from another perspective, etc. These new approaches are erasing a lot of that redundancy by saying at the start, let's identify what the relevant proteins are, and then go work on those in the more traditional sense. So that's a real change. And then in terms of the blueprint, I firmly believe that when we do figure out this blueprint of the cell, it really is going to be quite complicated network kind of property. And what I mean to say by that is that you're not going to be able to think about it again in terms of an engine of what's connected to what. Rather, it's going to be an engine where over time things move around and connect to something else, just like you saw in the movie. Which means we're going to have to think about design and architecture and engineering in a very different way. And that's exciting, it's challenging, it's daunting, and it also means something about how you train students in the life sciences. It's going to be essential for biology students to learn more math, more computer science, more network analysis, more statistics. These are going to become essential parts of the skill set that are going to be used by biologists in the next couple decades. And that universities are working to try to figure out how to create the training environments and the curricula that do that for us, but it's clearly happening and it's making us redefine fields, and that's fun. Change is good and it keeps us on our toes. And that's one of the points about the future of biomedical science. The other one, again, relates back to this engineering. Once you've got robots doing the experiments, the student can use, or the researcher can use their time to analyze the results. And so one thing that's already happened is that research has always been labor intensive. PhD students spend 5 years in the lab working on their degree, and they typically are in the lab 70 hours a week. And that's just kind of normal. They live in the lab. And it takes that much time to work out a problem, get an understanding of something, and write a thesis. Today, more and more, the students are in the lab, but they're not doing all the experiments. They're being helped by robots. I mean, this was science fiction when I was in school. And that means they're spending much more of their time sifting through data, analyzing it. And the data is so complicated that, as I said earlier, one person can't look at it and understand it all. We need computational crutches. We need good models to scan that data and help not just pull out information, but in fact pull out knowledge that teaches us something. And again, that's this real convergence between information sciences, engineering, and biology. And the benefits, I think, are going to be the benefits that we all hope for, which is new chances to tackle disease treatment, both diagnosis, knowing what to measure that correlates with disease, and then new targets. If in fact the cell is this big network of things talking to one another, then it's not obvious which of those is the weak link. And these kinds of activities are meant to help us understand what are the right targets. And we're still at the start of just thinking about what that means. So if you haven't been back on campus lately, there's been a lot of construction going on. This is bad for those of us who try to find street parking because all the streets are blocked off. But this is one happy outcome. About 4 years ago, the Gordon Center was completed. This is a building that's 230,000 square feet, 5 stories above ground, 2 below. and it wraps around Carrar Library. So it really goes around the city block. And what's exciting is it has within it space for chemistry, physics, biochemistry, cell biology, and the Ben May Cancer Institute. And it's leading to, again, a different model for training students. It used to be that the chemistry department was in one building, biochemistry was in this one, and physics over there. And as a student, you didn't go into one of the other buildings. You stayed in your space. So you didn't know what the graduate training environment and culture was like over there. Students that are being trained now have a much different experience. They're being trained in these multidisciplinary environments where they talk to students and faculty and other units, and it's very normal for them to now go out of their comfort zone and talk to somebody else about what they're doing. And it makes them very good problem solvers in the lab because they're very comfortable knowing what they don't know and knowing how to go out and talk to somebody who does. In my own case, that's the research group that I'm fortunate to work with. It's a group of roughly 20 PhD students, which are half PhD students and half postdoctoral fellows. Students who've just gotten their PhD at another institution and then come spend a few years with us. So they're the ones who do all the work. They get all the credit for what we managed to get done, and they're really what make the job so much fun, being able to work with them and share in their success and their hard work. So with that, I'm going to stop. I might have gone a bit over. If so, I apologize, but it really is something that's a passion of ours, and what really gets us excited is when we think we're at the first steps of what's going to be a big change that ripples through the field. And I think this idea that the cell can be thought of as a kind of computer, a kind of information system, and one has to think about states and networks, hasn't quite caught on yet, but my own bet is that that's going to be the way forward for us to really get an understanding of cell function. So I'll stop there, and I'm happy to have a discussion and take any questions. Thanks a lot for your kind attention. Going on to try to make materials that are smaller and smaller and smaller. And the benefits, for example, are that nanoparticles can get across the cell membrane much more efficiently than larger particles. So if you want to do genetic therapy and deliver a gene to a cell, one of the challenges has been how do you get the gene into the cell? And we're learning that if you put the gene in a nanoparticle, you can get it in. So that's just one example of where nanoscience is bringing us a new chance to treat disease genetically. The question is, what are the concerns? There's this gray goo line of thinking, which is the little gray goo will get inside of us and take us over and poison us. And there are examples already of nanoscale material being an environmental pollutant. Asbestos is in part nanoscale and clearly a concern. My take on this is that all chemicals have some risk of being toxic or dangerous, and we've got a way, in terms of environmental approval and monitoring, of testing each new industrial chemical to see how safe or how unsafe it is and then dealing with it appropriately. I think about new nano compositions and new particles as just being another chemical that should go through the same kind of evaluation and not thinking about it as something special because it's nano. It's a new form of matter, and it should be assumed not to be safe or unsafe. It should be tested, and we should know what it does. So I would treat them like any chemical, but it's an issue, as you know, is quite complicated, and it's not always the case that policy is most informed by technical arguments. So.
At the beginning of your fascinating lecture, I was not a science student, but I was sitting here just marveling. It was a very good, very interesting.
Thank you.
You mentioned molecular engineering and I think you used the phrase a new school of study, or forget exactly how you phrased it, that Chicago may embark on. So would that be within the biology department? Or, there's not traditional engineering now in Chicago, is there?
That's right, there's not.
Okay.
So how would Chicago launch a molecular engineering unit, whether it's a department or a center or an institute. It's not yet clear.
Okay.
There's been a lot of work going on, and different models are being looked at, and there's a number of faculty committees and outside committees that are having some input on it. What is pretty clear is that it would not be connected to one of the existing divisions or scientific areas, because it cuts across so many of the scientific areas. So I'm not the boss, so I can't tell you for sure how these things work, but I would expect that it would be set up as an independent unit that has strong ties to chemistry and physics and biochemistry, etc., but not fall under the power structure of any of those, so that it really has a chance of developing its own identity.
Oh, that would be very interesting to see how.
I'm fascinated by your description of the changed environment for teaching graduate students in the laboratory. The old school was that you had to do your hands-on and first, otherwise you couldn't understand your experiment. But it looks like you can do many, many experiments at once and still be pretty sure your results. Now, who's monitoring the machine?
Well, so when we do these experiments, we build in lots of controls. So about 1% of those spots are loaded with control experiments where we know the answer should be yes or no. And we use those to detect whether something went wrong, and in effect monitoring the machine. And it's also the case that a lot of conventional prep work goes into setting up a new high throughput experiment. So by the time the student gets on the robot, they've spent some significant amount of time working out the assay, testing it, what are the limits of how it should be performed, how to translate it to the robots. So they've also got a pretty good feel of how it works.
Well, you must have, as part of your training program, then almost a mechanical engineering. The students must be learning, or at least some of them, how to design the machines, not just run the experiment.
Absolutely.
Yeah.
So in a group
That's important.
It is. And I try to emphasize in my own laboratory that we don't run it in such a fashion that one student is an expert in part A, another student is an expert in part B, etc. But we try to make sure that the students involved in a project are participating in all the phases.
I think it's very exciting.
Thank you.
What do you think is the time frame? Like right now you said like with the care plan now, which you're basically taking one piece out and seeing what happens with the automated process. When do you think you'll have a blueprint or some sort of computational model that accurately represents some sort of biological system? I'm not talking necessarily human, but a simple microorganism or a virus. Where do you see that?
So my own thinking is that within the next several years, 5 to 10, we're going to have good models that can describe what single-cell organisms do in certain cases. So there's a really neat model organism, Mycoplasma genitalium. It's an organism that causes some venereal diseases, in fact, but it's interesting because it's single-celled and it only has 500 open reading frames, which is kind of remarkable that you can make a living thing out of 500 genes. Nobody thought that was going to be possible. And that's the one that's getting a lot of work. Craig Venter, who was involved in sequencing human DNA and has since set up an independent institute, is directing a staff of about 100 people at this problem of trying to model and synthesize that organism. And I think that's going to be our first success. At the human level, I think what we're going to see are models that describe a subset of the activities. And they're going to break if you push them too hard, but we're going to try to start with subsets that look like they're internally regulated, describe those, and then build off of them. But my sense is before 2020, for sure, we'll have a number of models that describe some set of activities in cells. And I think by then, we're going to have seen a real revamping of the curriculum in the life sciences.
they're here.
Excuse me. You concerned at all about, you have this immense computational power and experimental power with robots. I don't hear anything about hypothesis-driven experiments, and for my own area, what questions are you asking?
Right.
And we're always afraid that, we're ending up as our computer jockeys, as we would say, that know how to churn out the data and produce models, but they have maybe not much realistic application to the real atmosphere and ocean.
It's a good question, and again, I didn't get into that piece of it. The question is, traditionally biology and the sciences have been driven by hypothesis. You have a question, you create a controlled experiment to ask and answer that question, and you learn something that responds to the question. What these high throughput technologies are really intended to do is bring us faster to the point of framing a hypothesis. And so with those experiments, you find a relationship between some set of protein activities and a phenotype that you're interested in. That doesn't tell us anything about how it works. And so that is then the starting point for hypothesis-directed research to understand the details of those pieces. So these experiments, in fact, don't replace the usual way we've studied biology. What they do is they uncover the questions a lot faster than chance would have us uncover them in the absence of the technology. So that's the first point. The second point, though, is that I do think, and we already are seeing examples, that there will be global architectures that hypothesis-directed experiments will never reveal, because again, we're building from one node out. A global architecture, so to get a little bit more technical, because I know you're in the field, we have 500 kinases. None of them are specific. They all have overlapping specificities. And the network question is there something special about that set of 500 somewhat sloppy kinases? Can I take any 500 sloppy kinases? Or is there some more delicate relationship between what the magnitude of the overlapping specificity is that gives it the robustness? And those kinds of questions, they're network questions, and I think they can only be answered with these global analyses, not through hypothesis-directed research of the usual form. So I think there's going to be another set of questions where these are the right tools, but in other cases, these are just the starting point for the kinds of things you suggest. Tim?
Yeah, I wonder, Milan, you talk a lot about the impact molecular engineering might have on biomedical research, but I wonder if you might touch briefly on impacts in energy research or materials science and those kinds of things.
And this has been a big kind of point of discussion on campus. Where are the special projects, the special problems for molecular engineering? And the life sciences and medicine are clearly one. This is one shade of an example. There are other ways one thinks about this. But energy. I mean, one thing that's happened in the last couple of years is the nation is really renewing or creating anew its strong commitment to energy technologies and energy research. And energy is fundamentally about how do you convert one form of energy to a more useful form? How do you convert solar to electrical energy that's stored in batteries? How do you convert wind into electrical energy or chemical energy? And much of that happens with catalysts. Much of it happens with devices that are basically layered into nanometer layered sandwich structures. So molecular engineering, clearly one of the big areas for it is in energy science and energy technology, creating better conversion, better storage, better utilization. Another area that is very important are materials. One of the themes are multifunctional materials. We normally create materials that are optimized for one property. It's a good conductor, it's a resistor. It absorbs light well. It reflects light. It has magnetic properties of a certain kind. But what's happening now in material science, the kind of wave, is how do you create materials that have two properties? It conducts electrons in this dimension, but heat in that dimension? How can you create materials that are switchable, so that if I flip a switch, it was a conductor, but now it's magnetic? And if you had materials that had these kinds of multiple properties, that again would open the toolbox to creating technologies that do things that we don't even think about today. That's molecular engineering. Materials have function that stems from their molecular structure. So if you want to create a material with new function, it's really at the molecular level that you do that kind of engineering. So that's an area for the enterprise. And then there are others. So the challenge with molecular engineering is, where do you start? Because it cuts across so many areas. But that also means that you can be strategic about recognizing where your strengths lie, what can be recruited to campus, what the resources are, and pick the starting points and grow it from there. But this is going to be a broad field, I'm sure.
I had two questions. Is there a good understanding of the molecular basis of our body's automated repair systems? So, for example, if we cut our fingers with a kitchen knife or something. And second, what are the problems with three-dimensional time-dependent modeling of, say, for example, those kind of processes?
So the question is, do we have a good understanding of regenerative processes, wound repair, or organ repair? Short answer, no. We've got some understanding, but this is also a hot topic because of the tissue engineering and regenerative medicine angle. Instead of treating disease, could you just replace the organ? There's remarkable examples. Kidney. If you take a rat, you can literally cut out half of its kidney, and it will regrow that kidney in a period of 5 or so weeks. So that's an amazing ability to regenerate a tissue. Many animals, we know that they can lose a limb and regrow the limb. There's a form of a worm. You can cut that worm up into slices, 50 slices. Each slice will regrow into a full worm. So in biology, there are many examples of almost a freakish ability for organs to regenerate their parts. And we know that children can do some limb regeneration that adults lose the capacity to do, fingers, for example. So it's not that humans are not able to regenerate, in principle able to regenerate their parts. It's that evolutionarily we've not had to. There's not been an evolutionary pressure to do it, so we've turned off those mechanisms. But the thought is that if we could manipulate gene expression patterns in the appropriate way, we would impart an ability to regenerate organs. And this is also where stem cells play a role. If you take a stem cell, that cell has the ability to turn itself into one of many different types of cells. And so a lot of research is going on and trying to learn how to take a mature cell, a differentiated cell, and make it de-differentiate into a stem cell so that it can start differentiating again into what you need. So this is a big topic, a lot's going on, but it's very much still an unsolved riddle. But a lot of the work is happening in these model animals where you see this kind of robust regeneration. But imagine if you could regrow a limb. I mean, in principle, the blueprint might let you do that.
I want to leave you with one thought, because I was in a talk about this on campus last week, and there's evidently a faculty member who has an experiment where he boils an egg and then he unboils it. So I'll leave you with that to ponder. Imagine unboiling an egg. There'll still be dessert and drinks, I think, out in the foyer for just a bit. But I hope you'll join me in thanking Milan Mrksich for